Mm-hmm.
Yeah, great. It seems that
Mm-hmm.
recording.
Great. Great, thank you. And let me share. So, so can you see my screen?
Yes.
Mm-hmm. So, thank you everyone for coming. I see there are 26 participants. It's a world record for our mini conference, and I hope we will have more participants soon. And very nice to see people from all over the world, and especially from Ukraine as well. And just a short announcement. So first, I want to say that we have today great presenters, not just including me, of course, but other presenters are great, and I hope you will enjoy pretty interesting talks. And likely we have a very small, but at least we have some sponsorship. We have two Amazon cards with pretty small amount on them, but it's anyway kind of sponsorship. And the best talk and the best question from our guests will be rewarded. So I suggest um, to consider the following way of considering who will be the winner. As for the talk, the more questions will be asked for presenter will be considered to be the best talk. So maybe we will have to cut the number of questions, because who will start first, maybe we'll have the more questions, who will be the second will have just approximately, but who will be the last will have the last questions. So maybe we will just check the number of questions asked for a specific period of time, for example, for during the talk period. As for our participants, so I suggest that please ask your questions in the chat. Use chat for asking questions. I'll be moderating chat, and I'll be selecting the questions from the chat. The questions will be selected on the very simple principle, first come, first served. So because we have a limited time for questions, only like 10 minutes, so it will be only 2 or 3 questions may be answered. So if you have any questions, please try to answer them immediately using chat. So just write down your question into the chat. And please put any emotions to the questions you like, and the question which will get more emotions, emotion reactions, will be considered to be the best and will get his reward too. So that's it. Great. And I guess we may start. Time is running, and we have a limited time for presentations. And I would like to start with my presentation. And I would like to start it so with the words, I want to say that our mini conference is aimed for supporting Ukrainian science, Ukrainian neuroscience. So currently, because of war, our country is not very supportive for scientists, and times are still hard to do science, to run experiments, to run analysis, to write articles, papers, and so on. So our mini conference is a kind of a way to support neuroscience in Ukraine. And the first thing I want to say that I cannot omit it. So let me start with the presentation. I want to say a few words about our colleague, Ihor Zyma. Ihor Zyma was I can't say he was a friend of our department of Volyn National University Department, Human and Animal Physiology Department in Volyn National University. He was more than friend. He was a teacher. And Ihor Zyma was killed with his wife on the first day. It was actually the night, the holiday night, on the first day of 2025. I cannot omit this, and I want to say that Ihor Zyma played a great role in contributing in development of Ukrainian cognitive neuroscience. He was a person with encyclopedical knowledge. You can always ask him about any advice about experiment design, about data processing, about interpretation, and he played a great role in the development of EEG experiments and cognitive neuroscience experiments in our lab. So I want to say that we always will keep memory about him, and the seeds he sowed in Ukrainian science, I hope, I believe they will flourish, they will grow into big trees, and they will flourish and give great fruits, and that's maybe the best way to remember these great scientists. So actually, I want to present study which was inspired and which was discussed with Ihor Zyma. We both shared a great interest in connection in network models of the brain functioning, in analyzing connectivity in brain. And we talked about this study. So he was inspiring few ideas which I'm going to share with you today. And this study actually is being running by my PhD student Natalia Pak. So she is a little bit busy today and cannot perform and give talk herself. So I'll be presenting the data. So it's just a pretty fresh study. It's just on the phase of data processing. So a lot of things should be still figured out and sorted out. But anyway, let me start. I would like to start with some results we received in our lab few years ago. It was actually a paper published in 2018, and these results were related to decision making. So we used a protocol based on game theory model, so-called stack hunt, and actually it was aimed in studying altruistic behavior. And we recorded EEGs, processed them, and using low-resolution tomography, detected specific brain regions which were responsible for most prominent activity during decision-making, during altruistic decision-making. And what we got was a little bit surprising, because we got a very good coincidence with the regions which were described in literature by other scientific groups. So we got the same activity, the same type of activity based on our EEG results as the results which were obtained by other research groups using, for example, fMRI technique. But what was specific, that this activity didn't raise immediately. So the brain regions which sound, they became sound not immediately during stimulus presentation, as could be expected. but they did sound just on the time range 1 second or even later after stimulus presentation, which is not common, which is somewhat non-specific for brain recording technique for ERP, event-related potential technique. And that was pretty strange and pretty interesting because that means that actually in our paradigm in technique we usually consider the time intervals which we usually analyze less than 1 second. So it's usually considered that during when, for example, you get a stimulus, for example, like a green circle or something like that, then your brain responds immediately. For example, if this green circle has some meaning, you should press button when the green circle is presented to you. Then your brain acts very quickly. It takes like 200 or 300 milliseconds, so it's 1/3 or 1/5 of a second to create a response, to push the button or to withdraw your finger from the button, for example. So things go quickly, but anyway, we have two types of electric response of the brain. First one is very fast. It's called evoked response. So it goes immediately after we show you a stimulus, and then your brain reacts with changing with some specific brain potentials, which can be registered using specific technique like EEG technique, for example. And another type of activity is a little bit takes a little bit longer time to manifest. It's called induced response. And induced response usually is represented by oscillating activity. So there is a rhythmic activity. It means that after some time, usually it takes like second or even more, some rhythmic activity can be registered. So it's not like simple wave, it's like a series of waves, a packet of waves. So for example, these can be represented in here in this example of brain activity recording. So you may see that a very fast response, electrical response, goes immediately after presentation. It's of stimulus. It's like 100 milliseconds. However, then the activity, another type of activity reveals it's a rhythmic activity. And it reveals on a little bit longer time periods. For fast oscillations like gamma, it goes in 300-500 milliseconds. For slower types of brain activity like alpha rhythm, it goes within seconds and so on. So what we hypothesized is that effects which we had in this previous experiment were related not to evoked response, not to immediate response, but to rhythmic response. So it means that some kind of brain rhythmic manifested, appeared, developed, and then this type of activity manifested in the brain areas which were known for being active during, for example, fMRI studies. It was a little bit intriguing because this meant that fMRI studies and fMRI results may relate not to immediate response, not to immediate processing of the information, of the stimulus information, but to some activity which is evoked by stimulus, but this type of activity is pretty consistent, and this type of activity remains for a relatively long time, for a few seconds, and manifested in not immediately, but again in the range of a seconds. Actually, this idea is not new. And the idea of evoked and induced activity was suggested by, well, it's not pioneers of EEG technique, not pioneers of brain electrical activity recording, but scientists which have a pretty huge weight in the field of EEG studies. And so like Furtscheller, I apologize. And they suggested that induced activities so may be described by interaction. So I will use as a pointer. So the induced activity may be described by interaction of brain areas. And this type of interaction is cyclic. So these interactions, they form loops, and the activity goes up and down to cortex, then to subcortical nuclei, repeating and reverberating, and thus producing oscillatory activity. Not simply oscillatory, but rhythmic oscillatory activity. And this was the main goal, the main motivation for our study, which we are talking now about, that can we somehow distinguish evoked activity, and induced activity? Can we distinguish between activity which goes immediately after stimulus presentation and activity which reveals itself after a few seconds? The main idea was to study to use EEG recordings and to use these EEG recordings to first study what we get from the surface of the head, what signal we get. Then using specific techniques which allow to localize the sources of brain electrical activity, to find brain areas which produce this type of activities, and then find or try to describe how these brain areas are organized into networks, how they communicate. And in this way, we expected to check how induced activity and evoked activity are developed in the brain. So just a few words about technique we use. We use event-related potential technique. Event-related potential is actually recording brain electrical activity from the surface of the skin. So it means that if we put an electrode on the head of a subject, of a participant, then we register some kind of mixed activity which comes from different brain areas. And the issue with brain electrical activity with these induced responses is that they are pretty small in amplitude. So they are not very large. And ongoing, they are hidden in ongoing electrical activity. So our brain always does some stuff and produces ongoing electrical activity. And this ongoing electrical activity is usually 4 or 5 times larger in amplitude than specific response which is produced to some specific stimulus. So in ERP technique, thus we have to present same stimulus few times. And then we take I apologize. Then we take recordings of the signals which follow immediately stimulus presentation, and then we average them. Thus, we can exclude the ongoing activity, which is pretty chaotic and pretty occasional, and amplify the standard response. So the standard response is usually what is called event-related potential, and event-related potential is actually an evoked activity, activity with very a very short time of response, which is developed during first 200 or 300 milliseconds after stimulus presentation. So to test our hypothesis, and we designed an experiment. If you are familiar with attention studies, then you maybe will find this experiment design like pretty familiar. Because it's actually some kind of modification of attention network task. So during attention network, we present a series of stimuli. Each stimuli is a kind of picture on the monitor. And we start with fixation. There is a line, and our participants has to look at the line. Then we show the arrow. It means that arrow shows the direction where the main stimuli will be represented. The main stimuli then is represented by this graded circle. And this graded circle has two types of direction of the grades, from left to right or from right to left. And if graded circle appears on the side in which the arrow points, then subject has to push a button in a few minutes, in few seconds, when the screen with question your answer appears. So when subject sees question your answer, then subject has to press a button if the arrow was pointing into the direction of the graded circle. If arrow points into different direction, or if there is a distractor, another circle appears on the different side, then subject should withhold responding. So it's actually a pretty standard test, and we are interested on reaction for this stimulus. So all these stimuli are organized into what we call trial, and we have 120 trials. So it's a pretty boring procedure, and our idea was to make these stimuli which we represent during one trial to be presented in a pretty for a pretty long time, for 4 seconds or 4,000 milliseconds. So what's the point? So 4,000 milliseconds is not very common time for presenting stimuli in attention task. So it's pretty large time. So it's not psychologically rational usually, because it's too boring. You can just imagine that you are sitting and each stimulus appears in a very slow manner, and you are just sitting waiting for the next stimulus, and so on and so on. But given 4 seconds for each stimulus gives us enough time for analyzing oscillator activity, especially using connectivity measures, which usually need more time of the signal to be analyzed properly. And also it gives enough time for oscillatory response to be revealed and to appear in the EEG recording. So you may also say that this time when you was presented by stimulus, you have to wait for another stimulus, and it's pretty long time, 4 seconds, you get bored, you have some kind of ongoing activity like psychological activity, thinking your thoughts or something like that, and you're right. But anyway, we decided to give our subjects this time for perceiving the stimuli, and this was our main idea to check how brain behaves during this period. So I'm quickly skipping because we are short of time, some descriptions, but what we got. So first, we analyzed evoked potentials which we received from from the surface of the head. So it's kind of mixture of brain electrical activity. And you may see that we have these black patches. So this is actually the response to our stimulus. And black patches indicate the time when there was difference between stimuli when subject had to respond and when subject had to withhold from response, had to inhibit the response. And you may see that differences we have relate to first second mainly. So, and we almost have no differences in oscillatory activity, which follows after 1 second and lasts till 3 seconds. To get better analysis and better analysis results, we also used a technique which is called event-related spectral perturbations. So it's a kind of spectral analysis of the potential which we get, of the response which we get. And you may see that there is a difference between conditions when subject had to respond and subject had not to respond. So which can be, for example, found in the frequency range of 17 hertz. And these differences follow, for example, or can be detected at the time range of 1500 milliseconds. 1500 milliseconds. So it's actually a manifestation of oscillatory activity, not like evoked activity, but induced activity. And we also run analysis of brain areas which are actually representing the sources of brain electrical activity. We use specific techniques to locate them. Actually, one of them is independent component analysis, which allows to solve the inverse EEG problem, the inverse EEG task, and to find the sources of brain electrical activity. And we got main sources in the frontal areas, or in the frontal part of the brain. So here are they. And each source also can be described in terms of how this brain area responds on different stimuli. And you may see that we have some brain sources. Marked by red are the sources which contribute to the early response. You may see that the difference between two types of responses for stimulus to press button and stimulus to withhold from button pressing. So these red marked responses, they contribute to early processes, to early responses. But also we have some responses which contribute to looks like oscillatory induced activity, and they reveal they are sensitive to the condition, psychological condition, and the sensitivity is pretty stays pretty far in time from the stimulus presentation. It goes after 1 second, and between 1 second and 1 second and 1 and half the second. And there are also sources of brain electrical activity which also contribute to both early response and late response. And we used a connectivity measure which is based on Granger causality. Granger causality is a technique which allows to detect how one source of brain electrical activity influences or impacts another source. So it's a kind of effective communication between brain areas, and using Granger causality measures, we can detect which brain area pushes another brain area to respond. So, or makes some kind of impact on another brain area. And what we got was specific connections between brain areas. And you may see that, for example, brain source in right temporal area so was pretty sound, but depending on different conditions and depending on different timings, it was differently involved in brain connections. For example, if we have a condition when subject had to withhold from pressing the button, so the source was pretty active, but you may see that connections with other brain areas were pretty weak. And in other conditions, it was it had pretty strong connections with other sources of brain electrical activity, which means that brain networks and brain areas are organized in networks which change their structure and change their functionality during different time course of response and depending on a specific condition. So they are both time sensitive and condition sensitive. And you may see that there is, so we have found a paper on fMRI studies on attention network test, which is actually a pretty similar test to the one we used. And the structure of the sources of brain electrical, of brain activity are approximately the same, but these are fMRI study results, and this brain on the left represents our results. Short conclusion. So the main takeaway from my talk is that really we do have difference in evoked and induced activity. These evoked and induced activity are a manifestation of different neural mechanisms, of different brain structures activity, so which is not surprise, of course. But we assume that the differences in the type of activity during evoked response and induced response may relate or result in different metabolical changes and metabolical processes in the brain tissue, and thus what we have, the activity of brain areas which we have in later time course, which follows like 1 second after presentation of a stimulus, may significantly contribute to fMRI signal. Sorry for taking so long, and thank you, and stand with Ukraine. Thank you. So I'm out of time, and so there will be no questions. I will respond them in the chat if you have any questions. And we have another presenter. So Sergiy Yakovenko, who is a professor at West Virginia University. So far I understand his main interest is organization of movement. And he kindly agreed to present today. He has a pretty interesting studies related to computational approach in modeling brain functions, and I hope that you will enjoy his lecture. So Sergiy, thank you for coming, and the floor is yours.
Thank you so much. All right, it was a good start to diving into the neural computations. And do you see my screen at this point? Anybody? Yeah,
Sure,
fantastic. Yeah. So just a little bit about myself. I was born in Kharkiv, back in the Soviet Union. Then I actually moved to Lutsk for a couple of years and really
great.
Running along Styr and exploring Lundberg's castle. Then got back to Kharkiv, then lived in Prenzlau, Germany, then back to Kharkiv, then finally went to study back in Edmonton, Canada, Alberta, and followed to postdoc in Montreal, also in Canada, Then had a short postdoc in Berlin and a short postdoc in Pittsburgh with Doug Weber, who is going to present just a little bit later. And finally settled here in Morgantown, just a bit south of Pittsburgh, but a couple of degrees warmer down here. So it's been a bit of a trip, and we'll see what I have in the future. But I've been always interested in the way our nervous system is producing all these amazing actions. How do we control complexity of our body is still unknown question. And now, potentially more than ever, we really need to figure out some of the principles that can be then applied to solving hard problems like prosthetics. Limb prosthetics in particular is becoming a huge motivation for international community, and of course, very relevant for Ukraine. Here in Morgantown, we are working on several clinical techniques like osseointegration here. I hope you see my cursor. The idea is to implant a kind of a, it's not rocket science. Essentially, it's a small metal piece that goes into the bone, and it provides an anchor for the prosthetic to be connected directly to the skeletal system. Just yesterday, we worked with a patient who described this to be, the next opportunity that, because the prosthetic leg feels like your own leg. Another technique is called targeted muscle reinnervation. The idea there is not to just have a skeletal system modified, but also take into account that you have nerves that if you don't do anything about them, they potentially develop neuromas, and the limb develops all kinds of painful sensations. So here with Jack Gelman, we actually put these nerves, reimplant them back into the proximal muscles, and then they become an interesting opportunity for tapping into the nervous system to get additional signals about now, in this case, control of the ankle that is now not there, right? So if we have a prosthetic leg that has powered knee, powered ankle, we actually have access to all the necessary control signals. And this is just a current result from the lab that the video doesn't want to play. Oh no. Okay, that's not great. But so what you would see is a bilateral amputee walking on the treadmill in our lab. And this work, of course, you will see important people here in the corner. These are the students and postdocs that were working in my lab, and also colleagues who did all of this work. So we have to deal, with lots of structures that are hierarchically distributed, from those in the cortex that could provide interesting opportunities for implantable systems. As you can see in the examples here, this is a growing area of interest with for all kinds of devices that are invasive and provide long-term opportunities for access to neural pathways. We have a lot of interesting structure in the spinal cord. Now, in the spinal cord, we can also now embed systems that read from the peripheral signals as well as implanted signals in the spinal cord, and interfacing with the devices that are external. And of course, nerves themselves provide plenty of opportunities to embed devices into them, and you will hear more about this today. And that those devices could not only, of course, read information about the intent, but also we can use them for providing information back into the system. So using sensory feedback stimulation approaches that could encode sensory information. Now, structure is complex, but function is also pretty complex, and we need quite a bit of understanding of control systems to figure out how this all works together. One of the approaches is, ideas about dimensionality reduction. We've heard about them just from the previous presentation. And this is an illustration here how if we apply a concept that neural function is a dynamical system, we can visualize in more simplified space what that neural activity looks like. And in this case, dynamic system is supposed to produce a rotation within the JPCA space. It's a way to look at dynamical properties of the system. If you just put electrodes in the brain of an animal and a human doing reaching movements or walking, what you see is this interesting, almost like a wave propagating within different areas of the motor cortex. And this is my own work as a postdoc with Trevor Drew in University of Montreal. So you see kind of activation of some muscle groups initiating movement and then progressing towards execution of the movement. In this case, this is cat reaching and pressing on the lever to get a reward. It's almost kind of like a language of sorts. And one of the ideas that we would like to pursue is to see if we can use some of these large language models to have a generative approach to describe neural activity. This is something that I will not talk today, but we have people in chat here who work on this. And then, the function here can be also visualized together with the structure. So in this illustration here, we are playing activity that you see kind of, you can record activity from the muscles, not only from the cortex. So recording from the periphery, but then mapping that on the structure of the spinal cord. And then you can see spatiotemporal evolution of excitation within the spinal cord. And this is a cat walking, so there are 2 steps, and activity starts in this high lumbar region and then jumps to the caudal region and has certain profile. So behaves very much like a dynamical system, but seems to oscillate within the spinal cord. So lots of evidence that we have dynamic system working here. And that dynamic system is constrained by structure that it tries to control. So in this case, we're talking about limbs, and limbs are, they're segmented systems, but they also have certain mechanical preferences. And we know that from physics. We can just use equations of motion and see that this forward movement is a preferred way for our limb to work. Things to the side just don't work as well, or require extra effort. But this front movements is something our limbs are made to do. And interestingly, this is also illustrated here in one of my favorite papers by Tim Lillicrap and Stephen Scott from Canada, how you can look at the mechanics of the limb and see these preferred directions of movements, and also at the same time see the same preferences and the same shapes within the neural pathways. So the motor cortex knows physics, in other words. So you see these preference directions in the firing profiles of the cells, and these are just magnitudes of cell firing in these different center out movements. There is a connection. There is connection between structure that we try to control and also neural activity that controls it. Makes sense. And but requires a lot of computations. So basically idea that the brain knows physics potentially better than we do. So it does pretty sophisticated computations of our limb movements, yet things can go wrong. And one of the problems that we will all face, unfortunately, is aging. When we look at the aging as the parameter, what happens? The first thing that happens that you can notice that your parents are kind of slowing down a little bit. It takes them longer to respond to you. Reaction time goes down, but also their movements slow down. So there's this evidence comes from reaching movements as well as walking. So on the left here, the same center out movements that I showed in monkeys, now humans do the same things. And you can, looking at the age of the participants on the down here and time on the y-axis, you see that the people slow down. These are upslopes. It takes longer for you to make movement and also to plan movement. So those computations are slowing down. Same happens with the walk-in computations. And now these are this is like speed of the walk-in goes down, so we slow down physically, and that corresponds also to potentially planning for the steps and execution of the steps. So what's going on? We have structure and function interacting together to control complex limb movements. How does it all come together? There was a pretty old idea by Plato that we do construct reality kind of from almost limited sensory information. So there's an idea of the sort of sensory cave that we live in, and everything else, everything we experience is a construct, a model. And we use this model to do actions in the world, right? To control things. This was more recently expressed as the idea of the control system that embeds the plant that you want to control. So you would embed the dynamics of the limb or dynamics of any complex structure in the control pathways, so that you can predict delays, you can predict nonlinearities of that system. And generally it's called the Smith predictor, works for multiple applications. So this is one of the ideas in neurosciences, why we have complex computations. We could have some inverse dynamics computed, forward dynamics computed within these pathways. And that's the physics that previously Tim and Steve were showing, working in the cortex, as well as us thinking that these computations are prevalent in multiple pathways. Another example of what we compute is the dynamics of the body, right? So this is a beautiful illustration of a robot here, right? That is completely unactuated. So let me play this one again. So it has no motors, but it's walking and it has knees. This concept is called passive walking. And the idea is that we're actually built to produce this dynamic behavior, and it's in an efficient way. When we looked at the activity in the spinal cord, what I showed you before, we see the same oscillations now that mimic that mechanical oscillations of swing and stance of the robots, but now in the neural pathways of the spinal cord, you see oscillations corresponding to flexion extension, supposedly to control exactly the transitions to phases, to be more efficient and predictive about the walking patterns. And furthermore, we can build the whole representation of the arm and then use that to decode activity that otherwise is not so simple to understand. So here in this example, there is a whole scene. So what happens behind the curtain is that we have all these models inside and doing computations from muscle activation all the way to movement. And these are, of course, the people who did all the hard work. And now example here, Matthew is just showing how you can take Maya band and you can see signals floating from it. And now you run simulations of the physics and you're able to visualize the intended activity. And he's not even looking at the screen, which is kind of amazing. So you can have a fairly complex control if you have more sensors. And ultimately, the better the model is, the better the decoding is. So matching though the properties, dynamic properties of an individual to the signals is an obstacle, and we can find a way to further improve this. So this approach works. Embedding dynamics works, And that's kind of the insight for us to think about control of prosthetics this way. So here now it's a collaboration with the University of Pittsburgh, and the subject here has implanted EMGs in the stump of the limb, and now we're running these simulations. Well, we're running simulation that processes these inputs and interprets into the movement of the robotic hand.
Flex your wrist in.
So the key point here is that this subject did not have any time to train. So this is really immediate control, very intuitive, just thinking about opening and closing the hand, moving the wrist, and that's sufficient for the control of the prosthetic. Intuitive and just in time. So this idea of embedding of the computations is sort of comes all the way from my doctoral work, where I thought of representation of homunculus, in this case felinculus, within the spinal cord. So the organization of the spinal cord seemed to have this specific postural almost components that are there. And more recently, we examined this across species and saw that this is a repeating pattern in all the way to humans. And exactly what is embedded in the spinal cord for movement, it is a property that you've seen in the previous slide, where you have these oscillations controlling phases. That's one of the hypotheses. And it also has a history of people suggesting that this property exists, going back to Thomas Graham Brown, and idea that you have representation of these dynamics that helps you control limbs. The next thing is, well, because we are computational neuroscientists, we want to have model of everything, and what we can simulate, we can control, as you have seen from this idea of embedding. And the simplest model comes from kind of like thermodynamics field almost. It's mathematical biology, where you can represent the transformation of the CPG as a function that receives certain inputs, and we know what it's supposed to produce as the outputs. And this is some of the work that I did with Arthur Prohaska in the University of Alberta. So you can have this nice model, and then you can simulate it, and you could also just make it as a mechanical system. So out of yogurt cups, it's essentially two integrators with leaky components in them. So I have little holes here, and what you see is that this input signal, which is integrated by the yogurt cup from Germany, is producing nice oscillations. And you can even change the descending drive to increase the speed of oscillations. So, pretty simple idea. The math might look more complicated, but reality is that you can kind of run thought experiments and realize what the spinal cord is doing in these computations. And as you've seen, you can actually have an increase in input that changes and in time and produces appropriate modulation of flexor and extensor phases. So if we plot cycle duration versus phase duration here, the flexor phase, which corresponds to swing of the limb, doesn't change as we change the speeds, but it's the extensor phase that changes a lot. And this is something that we need to have in the prosthetics if we want them to work efficiently and also effortlessly. So in this case, if you go from right to left, the speed is increasing, and at the same time, what you see is as these phases get modulated. And actual simulations are these color points, and the black line is experimental data from cats. So pretty good match. So what we can do, we can kind of think about, okay, we have these relationships. We can describe them as the function of the CPG input, because we would actually like to know what CPG input is. And there was another nice paper by Goslow back in the day that connected cycle durations to the speed of the limbs. And when you do this transformation, turns out that the CPG input in the model seems to be linearly related to the speed of the limb. So maybe what we do is that we actually have a descending drive corresponding to the limb speed, and we control limbs. Based on that, we can build self-paced treadmills. And there's a little bit of diagram, but basically there are tasks where you can walk in VR, and you can start turning. Actually, I do have a video of this. So this is Matt's work. So in this case, he's trying to score. He goes to the ball and changes the limb speeds, and the treadmill adapts to him. And then you need to like collide with the ball object and try to score. So this game is under development. There's hopefully going to be a better character goalie, and the cool thing is self-paced omnidirectional treadmill that is enabling all of this. And the idea behind it is just taking that CPG model and kind of embedding it into the robot, which is the treadmill here. The treadmill is behaving kind of like it listens to your limbs and appropriately changes speeds of the belts and also VR experience of that. Now, how can we break this? Well, that analytical model is not interesting from the point of view of neural complexity. It doesn't really have many neurons. So one of the things we wanted to do is to build kind of a network of neurons using some of the simplest principles. And one of the simplest neural descriptions is coming from Eugene Izhikevich, and it's called also sometimes Izhikevich neurons. It's very simple. So this is the whole notation here. It's MATLAB description of it. But what's interesting is that it's able to produce a lot of different functions. It can spike in bursts. It can be statically changing activations. It can express quite a few, about 13 different states, I believe. So using a more modern tool called Nengo from Ottawa group, we, together with Yuri Prima, we built now a CPG model that used to be analytical. Now we translated it into the neural net that has spiking of the cells. And what you see here in the animation is actual population of select group of these spiking neurons that produce exactly the same cycling and phase modulation as you've seen in the analytical model. And so here we basically encode this spiking function to represent input-output relationships. And we can just take equations from our ordinary differential equation system of equations and just build them into the spiking model as a type of training. So it really learns those transformations. And then we can take the model through the paces, see that it's changing speeds, that it has description of the phases, just like we've seen in an analytical model. It does also recreate this limb control idea that the input is a limb speed into this model. The interesting part now comes when you start damaging it, right? So now you can just have all kinds of games with it. We can start killing these neurons in flexor pathways, in the extensor pathways, or all together, and see how the model responds to that. So what we've seen right away is that when we plot the damaged neurons as a function here, or error as a function of damaged neurons, error in the performance, as we start killing, progressively 1%, 2%, 3%, the function deteriorates and error increases here. And there are, the surprising thing to us was that different pathways are responding differently to the error. The flexor pathways are more resilient than extensor pathways. There's, when you kill neurons in extensor pathways, the model deteriorates quicker. And usually what that looks like is that you get kind of these missteps. So here's an example where the extensor pathways were affected, and you have this prolongation of failure to generate a step. And you can see these types of failures in actual cat recordings when it's called fictive locomotion, when we place electrodes in the spinal cord of paralyzed cats and actually look in the isolation how the spinal cord works. So this interesting thing that generally the pathways, even though they're structurally built in the same way, but function, once you train them to execute certain function, the error or the damage is differently affecting these pathways. So functional identity matters. But another surprising result was that we can save the pattern if we change the input. So here, when we plot the number of damaged neurons as a function of the CPG input for extensor damage and the flexor damage, the color indicates the error itself. And you want to have more of this yellow. That means the errors were low. And as we increase the damage here to 2%, extensors start failing. But at this point, we can just drive the system with an additional input, and we can get ourselves into the yellow area again. So we can save the pattern and improve the performance. And you can see this saving opportunity in both flexor and extensor pathways and altogether in the model. So what Ihor came up with, and there are some examples of these from different parts of this diagram, but what Ihor came up with is this idea of a rusty watermill. Idea that you can think of the damage, neural damage in these dynamic computations as a type of kind of if you think of the Dutch watermill and rusty axle that produces extra friction, you can just override it with additional water drive, right? So this is example here, or illustration of this. The idea that the slowdown in the computation can be just rescued by the additional input, up to a point, of course. And now going back to the aging question, well, maybe that's what's really happening. Our dynamic computations just get these additional errors that 1-2% of neurons are dying, and computations are less efficient, showing the reduction in the reaction time. And what we have to do is to just get an extra input to recover them. So this is the main idea of the talk, and I would like to thank my lab mates and also collaborators, and of course the funders. National Institute of Health started my lab pretty much. Then I was lucky to receive quite a bit of funding from Department of Defense, and currently we're sponsored by the Air Force to do some of this work. And just maybe shout out that it's really exciting to do things in the lab. Kids enjoy it, and you can also have some cool technologies with virtual reality that could help in rehabilitation of the wounded warriors, as well as could be really a lot of fun to play. Do we have time for questions?
So we're currently short of time, and I do not see any questions asked in the chat, but Sergiy will stay with us, so far I understand, till the end of our conference, so for a few hours more. So I guess I suggest to ask Sergiy questions either in chat or maybe after the conference. Is it okay?
Absolutely.
Okay, thank you. Okay, great. Very nice presentation. I definitely enjoyed it. And now I want to give word to Seonmu Lee, who is a postdoc at Purdue University. Seonmu works with fMRI studies, and he presented, it seems to me, a year ago, a talk at the local colloquium about attention and memory mechanisms. And actually, I did like this talk very much. It was very neat, very clear. I couldn't check the data. I do not understand much in fMRI data, but I definitely recommend to listen to the talk of Son Muli, and I'm pretty sure the talk will be great. Thank you. So Son Mu, so you may, the audience is yours.
Thank you. Yeah, I'm Somu. It's my pleasure to be here. Thanks, Illia, for inviting me. I'm a postdoc at Purdue University. Today, I'm going to talk a bit about my research, which is using functional MI to understand repetition and attention in memory formation. Okay, so there is no doubt that memory is one of the most important cognitive functions we have, helping us interact with the world. So my research interest and long-term goal is to understand how is memory formed in our brains. Think about studying for an exam. You don't just read a textbook once and magically remember everything. You need to review the content multiple times to form the memory. You also need to keep focus while studying. You just simply looking at words passively is not enough. So repetition and attention are two important factors for memory. In the following 20 minutes or so, I will talk about two studies on how the brain processes repeated information and how attention affects the brain process information. So how can we study repetition and attention in our brains? We use functional MRI, which is a non-invasive technique that measures brain activity. So here's how it works. When your brain processes a stimulus or performing a task, the neurons become active and start sending electrical signals to each other. To keep working, these neurons need energy. So they signal nearby blood vessels to send more oxygen and nutrients. As a result, more blood flows to that brain area. Since oxygen levels affect the local magnetic field, MRI scanner can detect these subtle magnetic changes. So what fMRI really measures is called blood oxygen level dependent or BOLD signal, which is an indirect way of looking at brain activity associated with specific stimuli or tasks. And one common way to analyze fMRI data is through univariate analysis. Here, a voxel is the basic unit of a 3D MR image. This method averages brain activity across voxels within a region, and then compares the average activity between different conditions or between regions. For example, we can compare brain activity when people moving their right hand versus they do nothing. The univariate result will show increased activity in the left motor area, or we can compare remembered versus forgotten items. It will show more activity in the hippocampus for remembered items. We can compare different conditions to know brain activity related to a variety of cognitive functions like object recognition, emotion, decision-making, etc. So you might guess how researchers study repetition effects. In a typical experiment, participants see a series of object images and make a judgment on each object, like if it is living or not. The judgment here is not that important, just to make sure they are paying attention. The important point here is that some of the objects appear just once and others repeat. Studies like this usually found reduced brain activity for repeated objects compared to new objects. This phenomenon is called repetition suppression and happens in the frontal and fusiform regions. But why does brain activity go down for repeated stimuli? Several theories try to explain this. You can also think about how to explain this phenomena. For example, fatigue theory thinks that all neurons get tired and respond less to a repeated stimulus. On the other hand, sharpening theory suggests that only the most important neurons fire again, so become more selective. And synchronization theory thinks neurons become more efficient by firing together, but with less overall activity. And predictive coding theory proposed that brain predicts repeated information, so it doesn't need to respond as much as the first time. So all these theories explain the repetition effects from different angles, and of course there is evidence for supporting each of them. So how can we test them? The key difference here is that theories such as fatigue and sharpening view repetition suppression as a local effect happening within a single brain region. In contrast, theories like synchronization and predictive coding emphasize the communication between regions, so repetition should affect the connection between them. So to test this prediction, we used another common method called connectivity analysis, which Illia just introduced. This method also averaged the signal within a region, like univariate approach, but considers the time information. So it measures the correlation between the average signal from different regions over time. And if it measures just the correlation, we call it functional connectivity. If it estimates causal or directional inference, like which area is driving another, we call it effective connectivity. And I will come to this in a minute. So in this study, we use an open access functional MRI dataset where participants saw phase images or non-phase noise images and made left-right symmetric judgment. We analyzed the signal from 3 phase processing regions, including fusiform phase area, occipital phase area, and early visual cortex. Then we used dynamic causal modeling, or DCM, to measure effective connectivity within or between regions. And DCM first estimates the fixed or endogenous connectivity. This baseline connectivity is how regions communicate with each other in general, regardless of stimulus types or conditions. Then it estimates whether and how this connectivity is changed by experimental modulation. So in our case, when there is a repeated image. Finally, we have to specify which region is receiving the sensory input. In our case, it's early visual cortex. Here is the result. The results show the probabilities of repetition modulation on each connectivity. We found strong evidence that repetition modulates connectivity between early visual cortex and occipital face area, and connectivity between early visual cortex and fusiform face area. So this result suggests that repetition doesn't just cause less activity in one brain region, it changes how regions interact with each other, supporting the theories like synchronization and predictive coding theories. Okay, so now let's talk about attention. And it is well known that the brain has two primary visual processing pathways. First is the ventral pathway. The ventral pathway is mainly responsible for object recognition, such as distinguishing a chair from one another, but also generalizing the concept of chairs across different shapes and colors and size. Another one is the dorsal pathway. Now, ventral pathway is from occipital to temporal regions, and dorsal pathway is from occipital to parietal regions. Dorsal pathway mainly process spatial information, so we can locate an object and interact with them. Okay, so the goal of this study is to see whether and how attention to shape or location affects both ventral and dorsal pathways. Specifically, how attention influences the way neural representations or neural patterns are organized in the brain. So we designed an experiment where participants perform a one-back task. During the task, they saw the 8 shapes at 8 possible locations. In one condition, they pay attention to shape. So pressing a button when the same shape appeared, regardless of its location. In another condition, they attend to location. So pressing a button when the same location is repeated, regardless of its shape. In the last condition, they just passively view the stimuli. Okay. And we applied multivariate pattern analysis or MVPA on our data. Now, instead of just looking at average brain activity, this MVPA method keeps the signal from each VOC source and directly compares the pattern of activity across regions or under different conditions. So that's why I said we aim to understand how these patterns or representations are affected by different attention conditions. Here is an example. Suppose that the activity in a region looks like this in shape attention condition for the first shape circle, and look like this for each shape. And the pattern looks like this in another condition. And if we just compare their mean activity, we may conclude that there is no difference between conditions. Also, we may conclude that there's no difference between shapes. However, if we calculate the similarity between the similarity of this pattern between shapes and plot them in a 2D space, you can even tell by your eyes that some shapes share more similar patterns, but others are more distinct. If we plot them under different conditions and also measure the distance between them, now we can found that shape attention might cause greater distances between shapes, meaning better discrimination compared to another condition. And we can do the same thing for locations, plotting the neural representations of 8 locations in a 2D space and measuring the distances between them. So this is one of the current results from just 9 participants, and we are collecting more data right now. Here we get the signal from middle occipital gyrus, which is in ventral pathway and supposed to process shape information, but we plot the representation of the 8 locations here. So the red dots are the 8 shapes under shape attention condition, and the blue dots are 8 shapes under location attention condition. And we can measure the distance between these 8 dots. Here's the result. We can see that in the shape attention condition, the distances of the 8 locations are greatest compared to passive viewing. So the result is quite interesting because it suggests that when people are paying attention to shape and in a shape processing regions, their brains also represent locations more clearly. Okay. To summarize, first, what happens in our brains when we see an imaging repeatedly is a simple question, but not yet understood. With MRI and connectivity analysis, we emphasize the role of interactions between regions during repetition. Second, our preliminary results suggest that attention can influence location representation in ventral region, which is so to process shape information. This challenges the traditional views that separate what ventral and where dorsal pathway. Lastly, and perhaps the most importantly, by showing the 2 examples of repetition and attention studies, I hope to give you an idea that how we can use MRI to understand what happens in our brains during cognitive process like memory. Finally, I'd like to thank my advisor, Professor Rick Hansen in Cambridge, Professor Anne Serino at Purdue, and some of the funding agencies. Thank you.
Great. Thank you. Really intriguing and really interesting. So we have time maybe for 2 questions. So I do have one question. So these results regarding like challenging the traditional view on what and where pathways is really intriguing and maybe even revolutionary. So far, maybe I missed something, but so far I understand you focus mainly on activity of occipital areas. And what and where pathways, they finish usually in frontal cortex. So far I remember. So the what pathway is considered to lead to dorsal lateral prefrontal cortex, while the where pathway is usually related to the activity of the frontal cortex, which is located more ventrally. So my question is, so your new data about the organization of what and where path, do they suppose, or can we suggest from your data that we will have some type of what activity in frontal regions which are related to where system and vice versa?
Yes.
Am I clear enough?
Yeah. That's really a good and important question. And I just showed the results from one region here, occipital region, but in fact we also look at other regions like parietal for where pathway. So the next steps, of course, we can look at more regions like frontal regions you mentioned. And I think, yeah, from our data, I would say that we might see the what activity also in where pathway, I believe. And I think another point is that how this activity is changed. Maybe in the previous years, maybe we didn't see the what activity in where pathways, that there's just no, like, their activity or representation is very stable and not affected by attention, for example. But now with this pattern analysis, we can see exactly how they changed under different conditions. So I think, yeah, it's possible. It's possible that where pathway also process some what information, I believe. Yeah.
Great. So no questions at the moment, extra questions. So, okay, I just want to remind that to our participants, so please post your questions in our chat to speakers, and don't forget to vote for most interesting questions. So I just want to recall that we have 2 Amazon gift cards, very small amount on them, but anyway. So which will be rewarded to the best question from participants and the best talk from our presenters. So just support the activity. Great, thank you for the very nice talk. And our next presenter is Douglas Weber. I'm very glad to have Douglas on our conference as our speaker. Douglas represents Carnegie Mellon University, and his research mainly focused on connection between motor and sensory function. And I think it's the most hard way to study neuroscience. So Doug, floor is yours. You're welcome.
Thanks, Ilya, and thanks for the invitation to present in this exciting conference. Sorry, I missed the first couple of talks. It's a busy time of the year. So,
Doug, just one more, I apologize. One more remind, if you want to stop recording, just let me know. So this conference is recorded and will be published on our channel.
Yeah, no objections. Okay, so I'll just dive right into it. First, I want to introduce my lab. So I co-direct what we call the neuromechatronics lab at Carnegie Mellon University. Darcy Griffin is the co-director of our group. And we try to work across disciplinary boundaries to converge ideas from many different perspectives, neuroscience, physics, different branches of engineering, computer science, and so on, all with the goal of achieving a deeper understanding of how the nervous system senses and controls movement, and then enabling the engineering of new technologies to restore function after neurological injury, such as stroke and spinal cord injury. So this is a slightly outdated picture of our group, but our beautiful city is shown in the background, and I invite anyone interested to come visit us, especially in the summer. Okay, so just to make people aware of where Pittsburgh is, so we're on the kind of northeast corner of the country, down in the southwestern corner of Pennsylvania. Within the city of Pittsburgh, we're fortunate to have a number of really top-notch universities and medical centers that enable us to work collaboratively on both fundamental neuroscience and engineering challenges, as well as applied clinical translation. And I just want to give a shout out to some of my collaborators at the University of Pittsburgh, specifically Marco Capogrosso, Vera Pirandini, Lee Fisher, George Wittenberg, and Jen Collinger, because I'll be talking about work that we're doing collaboratively with them, as well as our physician colleagues, Peter Gersten, David Lacomas, and Raul Noguera. Okay, so as I mentioned, we try to work kind of across the spectrum from basic science to last mile clinical translation. And fundamentally, we're interested in understanding the role of sensory systems in forming and shaping motor behavior. And as well, we try to understand how devices like neuromodulation treatments interact with those systems with the goal of improving motor output. We also develop technology, both hardware and signal processing methods to enable us to measure and understand neural function and build assistive technologies that link spared function to control of assistive devices. We do a number of early stage clinical trials, kind of first in human studies, evaluating emerging devices like endovascular brain-computer interfaces, as well as non-invasive devices like myoelectric interfaces that people with spinal cord injury can now use to interact with digital devices. And more and more, we're trying to bring these devices not just to the clinic, but to the commercial market. And so some of the work that we're doing involves a startup company that we formed a few years ago, Reach Neuro. And so it's really through partnerships with companies, both large and small, that we hope to really maximize our impact on society. All right, so what am I going to talk about today? I'll just provide really brief overviews of the work we're doing currently in brain-computer interfaces. I'll also highlight some of the work we're doing on muscle-computer interfaces. And then lastly, I'll talk about work we're doing using spinal dorsal root stimulation or spinal cord stimulation to boost motor function after stroke using devices implanted in the epidural space of the spinal cord. I won't talk about the work we're doing on transcutaneous spinal cord stimulation, but I'm happy to field questions on that if anyone is interested. All right, so I probably don't have to spend too much time on this slide. I think given all of the recent activity in the brain-computer interface space, I think most people understand that the concept is to record signals for motor areas of the brain, signals that are assumed to encode information about intended actions. From those signals, we can decode what those intended actions are, and then route them to devices that can execute those actions in service of the individual with a disability. And there's a lot of different technologies that have been developed over the year, developed and used over the years to measure various features of neural activity in the brain, whether they're local field potentials or single unit action potentials recorded with electrodes implanted in the brain, or signals recorded non-invasively using scalp-placed EEG electrodes. In all cases, the assumption is that there's information embedded in those signals that we can harness to operate an assistive technology. One of the workhorse technologies over the years is something called the Utah electrode array or the BlackRock electrode array, which works extremely well and provides very stable recordings from motor cortex, from single units in motor cortex over now decades. The challenge, though, however, is that it's a technology that requires a fairly significant surgery. We have to open the skull, open the dura, penetrate the brain tissue with literally hundreds of fine needles, all in the service of extracting motor control information directly from the brain. And an emerging technology that offers potential advantages for patients in that it doesn't require an open craniotomy, doesn't require opening of the dura, and really doesn't even require direct physical contact with brain tissue, is something called the stentrode, which is an endovascular brain computer interface that is delivered by a catheter placed in the jugular vein and then routed to the superior sagittal sinus to a location immediately adjacent to the motor strip. And by releasing the stentrode electrode array at that location in the brain, we can listen in on the electrical activity generated in nearby motor cortex to get a readout of what people are intending to do. And so this is what the stentrode system looks like. It's a 16-channel electrode array that is attached to a somewhat traditional endovascular stent that expands inside the blood vessel to make physical contact with the blood vessel walls. The leads from the electrodes are tunneled down through the jugular vein to a chest cavity that contains an internal telemetry unit. The electronics for the system are embedded in that internal implanted telemetry unit and then routed out wirelessly to a connector that takes the signals to a computer. The devices are implanted permanently, and patients can use the technology in their home. A few years ago, we started the trial in the US. This is not the first in human trial. A previous study was performed in Australia where 4 patients with ALS were implanted with the device, and the safety record has been essentially perfect to date. In the US trial, which includes University of Pittsburgh Medical Center, Mount Sinai Hospital in New York, and University of Buffalo in upstate New York. And Synchron is the company that is developing and commercializing the stentrode. So in the US-based trial, we've implanted a total of 6 patients to date. 5 of the 6 have severe paralysis due to ALS or Lou Gehrig's disease. One of the participants has severe paralysis due to brainstem stroke, and all have no or very limited ability to interact with digital devices. This is the team at CMU that's leading this work, and so I just want to acknowledge the work that I'll be sharing now. So one of the tests we do to evaluate signal quality from the stentrode is something called a motor mapping test, where participants, once implanted with the device, are seated in front of a computer and then prompted to make attempted movements of different body parts. Ankles and or feet and hands are the typical targets that we use. And when they're prompted to make those movements, we ask them to rest for a short period of time, then make an attempted movement, then rest again. And through those alternating periods of rest and attempted movement, we can examine changes in the signal properties to identify features of the signal that modulate with those attempted motor actions. And so if we just look at a couple of examples, here is data from our first 2 participants. And what we see when we look at the power spectrum for I think this is all of the channels that were recorded. what we see is that across a fairly wide part of the power spectrum, we see an increase or an enhancement in signal power, particularly in the low and high gamma bands, indicating that indeed there is modulation in these signal power during attempted movement. And so we can analyze those signals further by isolating low and high gamma band features, just using bandpass filtering and then smoothing to get the envelope of the signal, and then measuring the amplitude, the changes in signal amplitude in each of those bands as the subject alternates between resting and attempted movement periods. And so if we look at the strength of those signal changes, we see that on some of the electrodes, we measured amplitude changes on the order of 100 to 200%. So these are fairly large increases in signal power associated with attempted movement. That was for participant 1. In participant 2, we saw much smaller signal changes, but still significantly greater than 0, indicating that we can reliably detect events where the user is attempting to move. If we look at the stability of those motor signals over time, here we're just showing 3-month windows for those 2 participants, and we see that the signal to noise ratio for both low and high gamma band features looks pretty solid over this 3-month span. I will note that we'll be releasing a paper later this year reporting the results from all 6 of our participants. But the early analysis looks very encouraging. And in addition to measuring just kind of gross changes in signal amplitude related to motor attempt, one of the other things we're interested in understanding is whether or not there's any somatotopic organization in these features. And if so, then perhaps we can control not just a single motor output, but potentially multiple motor output channels. And so here, if we take the low and high gamma features that we measure in this motor mapping task, and we reduce them, if we project them down into a lower dimensional space using linear discriminant analysis, what we see is that there's distinct clustering within this space, indicating that the pattern of signals that we measure is somewhat distinct for each of the different movement types that we ask participants to perform. There's certainly a bit of overlap in these features in at least some of the subjects, but there is at least early indication that we might be able to discriminate not just a single channel of movement, but perhaps multiple channels. And then lastly, I'll just show a quick video here demonstrating one of our participants using this interface in a very practical and important application. So here he's filling out a pain questionnaire to report to his caregivers any presence and location of pain in his body. So this gentleman has ALS. He's locked in. He has no other way to communicate with his family members and caregivers. And so now with this device, he has that ability to express himself. So the takeaway here is that in this early feasibility study that we're doing in the US, we see strong indication that the stentrode is capable of measuring motor intention from the brain using this minimally invasive implant, and that the signal properties look to be fairly stable over time. And this paper just came out this week, I think, in the Journal of Neural Engineering, reporting the early results from this study, showing the data from our first 2 participants. And an expanded version of this will be coming out later this year, hopefully. Reviewers will be willing. So this work is using devices that are implanted in the brain. So surgery is involved, and so some non-significant risks that are that patients must accept to adopt a technology like this. And so while it may be a desirable option for many participants, we envision a non-invasive alternative might be desirable as well. And so to that end, we're also looking at developing wearable sensors that can be worn on the body to detect motor signals that are spared after diseases like ALS and spinal cord injury. So this is the team working on this project, a large group. Jordan Ting really spearheaded this project a few years ago, and Dalen Despradel and Max Murphy are now leading it. And so the work really began as an offshoot of our BCI studies, where we had participants with tetraplegia coming into the lab to do BCI tasks. And one day we decided, well, let's just see if we might find any evidence of EMG in their arm. And so we wrapped an electrode array around their arm that had 150 electrodes embedded in a flexible sleeve. I should note that this sleeve was developed by Battelle Memorial Institute in Columbus, Ohio. And what we found is that when we would prompt the users to make attempted movements with their hand, even though the hand didn't actually move, we still saw evidence of EMG being generated in their forearm. And so that gave us hope that if participants can still generate and volitionally modulate activity in muscles of the arm, that perhaps that might be a way for them to express intended motor output. And so that work was published a couple of years ago, but led to a new collaboration with Meta, who is developing a wristband EMG system for augmented and virtual reality applications. And so using the Meta wristband, we have participants with tetraplegia coming into the lab, and we're mapping the motor activity that is detectable with this wristband, and we map it out for different attempted movements of the hand. And so again, even though the participants can't move their fingers, we can use their EMG as a control signal for controlling movements of a cursor, as you're seeing here. So they're using motor unit activity to control the position of cursors on a screen. Here, the participant is playing a simple video game that we made to help the users train on how to modulate their activity for controlling cursor movements. And the video games also serve as good motivation because we keep score, and the participants always like to try to improve their score and compete with their peers. And so what we're seeing is that, even though the participants, can't generate the physical movements needed to move a joystick or type on a keyboard, they do retain enough level of control of their muscle activity to control digital actions on a screen. So here's a 2D version of a game that we created, so the subjects can control multiple channels of motor output using these motor unit signals. And so the takeaway here is that, not only can we use implantable devices to enable people with paralysis to control digital devices, but we can also use wearable devices as well. And so now the last project that I want to talk about is aimed at trying to really enhance the spared motor function that persists after stroke using a technique called spinal cord stimulation. And so this is the team working on this project. Nikhil Verma and Luigi Borda are PhD students in the lab that are leading this project. And so just to sort of set the stage for discussing this, I think it's important to appreciate what happens in the brain after a stroke. So if we simplify the cortical spinal tract this way, what we see is that after stroke, damage to the cortical spinal tract disrupts communication between primary motor cortex and the neural networks of the spinal cord that are responsible for generating and sensing movement. And so that loss of communication between cortex and spinal cord results in a number of deficits. Weakness is maybe one of the more obvious, but spasticity and disruptions to the coordination of activation across muscles also tend to be very disruptive in producing motor function. And so the approach that we're taking, I think, is a distinct departure from more traditional approaches to trying to correct the deficits that are produced by stroke. So a lot of attempts to date really focus on either stimulating muscles to boost activation of muscles that are weakened by stroke and spinal cord injury. Or an alternative approach is to bypass the muscles entirely and use robotic exoskeletons to generate an externally applied torque that can, again, further actuate the limb. But our approach is really to go upstream from these locations to try to see if we can improve the motor control functions that are still present but slightly disrupted in the brain and spinal cord. And so by targeting CNS structures rather than peripheral structures, our goal is to try to improve neural control rather than just treating deficits in torque production. And so our approach really leverages our understanding of spinal cord circuitry and especially reflex circuits in the spinal cord, and recognizing that primary afferent neurons, especially 1A afferent neurons, provide direct access to motor neurons that are needed to activate muscles to produce movement. And these circuits provide not only the ability to excite agonist motor neurons, but through pathways of reciprocal inhibition, we can also suppress activity in muscles that might be opposing movement, such as often occurs in the case of stroke. And so our approach is to electrically stimulate primary afferent neurons to drive excitatory and inhibitory inputs to spinal motor neurons. And so the technology we use is a well-established technology called spinal cord stimulation. It's been approved in the US for treating pain since 1987. And it includes an electrode array that is positioned on the epidural surface of the dorsal spinal cord. We position it at a slightly different location in our application because we're targeting the dorsal root entry zone. But in doing so, we can excite primary afferent inputs to spinal motor neurons to boost the excitability of motor neurons and enhance muscle activation. And so just to show you an example of how, oops, skipped over that. My animation's not playing. So one advantage of this approach is that it's a fairly minimally invasive procedure. So we can access the epidural space using a TUI needle that is wedged between adjacent vertebrae. And through the barrel of the needle, we can thread these electrode arrays that provide a set of contacts that are distributed along the length of the cervical spinal cord and provide several independent sites for stimulating neural tissue around the electrode. In our study, we're implanting a pair of these 8-channel electrode arrays for a period of 30 days to allow us to study the effects on motor function. So the system looks like this. The leads are implanted in the cervical spine, spanning the C3 to T1 segments. The leads are externalized and connected to an external stimulator and a custom controller that allows us to create arbitrary patterns of stimulation across the electrodes in the array. And so one of the first steps is to map the muscle activity that is evoked or facilitated by stimulation at each of the different electrodes. And we know that there is a fairly clear somatotopic organization to the motor neurons in the cervical spinal cord, such that if we stimulate at a rostral contact in our array, we'll produce activation that is primarily localized to the upper arm and shoulders. If we stimulate further down in the electrode array, the site of activation in the arm will move distally. And so we can cover both, muscles from the shoulder all the way down to the forearm to facilitate recovery of function throughout the arm and hand. So now I'll just show a couple of examples of the effects that stimulation has on motor function. So here, this split screen video shows our participant performing a box and blocks test. At the top, no stimulation is being applied, and you can see she struggles a bit to grasp and transport the blocks from left to right. When in the bottom panel, where stimulation is being applied in a tonic fashion to 2 electrodes in the array, we can see that her hand function and her ability to transport the objects is greatly improved, resulting in higher rates of completion. And then the last video I'm going to show shows the participant performing an eating task, probably my most important activity of daily life, or at least my favorite activity of daily life. And you can see here that when stimulation is off, the participant has a difficult time holding the fork in her hand. She gets hold of the fork, but her grip force is not sufficient to keep the fork from sliding out of her grip. And like many people with hemiplegia, she has to rely on her so-called good hand to help assist her affected hand. But eventually she gets a hold of the food, and because of impairments in her forearm and elbow, she can't bring the fork all the way to her mouth. She can't supinate her wrist or flex her elbow. And again, she has to rely on her good hand. So now a few minutes later, we'll turn on stimulation, and you'll see that her hand function is much improved, and as well, her forearm and elbow work better as well. So now she gets hold of the food with one try. She can supinate her wrist and flex her elbow to bring the food all the way to her mouth. So again, this is tonic stimulation. So we're not patterning the stimulation to support distinct movements. All we're doing is providing tonic facilitation of the voluntary actions that she's directly controlling. And so I'll just quickly summarize. So at the beginning of the talk, I showed you that we can use implantable devices in the brain, as well as wearable devices on the arm to detect and interpret motor signals that express someone's attempt to move, even when paralysis prevents them from generating those overt movements. And that if we use electrical stimulation targeted to sensory structures in the spinal cord, we can amplify spared motor function to improve performance of activities of daily life. And with that, I'll take any questions. I apologize for the race, but I really wanted to highlight some of the exciting work going on in the lab.
Brilliant and very exciting. So it's always a great thing to watch how people restore their functioning under specific treatment. So we have a few questions. Actually, I would like to ask one question from myself, but I see that we have a pretty popular questions. So I will post my question maybe just in chat, and that's it. So the question is, what is the time frame between a stroke presentation and device implementation? Is it possible to use your device if patient has MCA, both hemorrhagic and ischemic stroke?
So these are excellent questions. I'll address the timing question first. So the participant that I showed you was 9.5 years out from her stroke, so well into what we call the chronic phase of her recovery. We've only tested people that have been a year plus out from their stroke, and we see a range of effects that depend heavily on not only their baseline motor function, but also their level of spared sensory function. So really identifying the ideal sort of responder profile is still a work in progress, although we have some insights that we feel pretty good about. You know, we're focusing on chronic stroke right now because, well, for a number of reasons. Number one, they're neurologically stable at this point, and so we can be confident that any gains in function we see can be attributed to the stimulation and not their kind of normal or spontaneous course of recovery. We've had a number of conversations with stroke neurologists over the years that have led us to think that, you know, there would be an interesting opportunity to use this technology much earlier in the recovery, even during the acute period of recovery, to avoid the development of sort of compensatory strategies that might hinder someone's ultimate recovery. So it's way too early to say that, you know, yes, we should be doing this immediately post-stroke, but I do think that that eventually we want to intervene earlier than we're currently doing. The question about stroke type, again, this is a subject of debate within our group and within the community. I, for one, think that that this is a technique that can work for both hemorrhagic and ischemic strokes, subcortical as well as cortical. It really is going to depend on what level of spared function they have, because all we're doing is we're amplifying spared function. So if there is literally no measurable voluntary function persisting after the stroke, then I would be less hopeful that we can provide a significant benefit. But where there is a measurable spared function, I think this will help.
Great, thank you. So there was one question in here posted, but I do not see it anymore. Was it deleted? Okay, so in this case, I would like to answer the question just from myself. You was talking about phasic stimulation to improve or to restore the motor activity. So I just want to ask if there's some habituation effect to this, whether the efficiency of the stimulation decreases with some time because of some kind of habituation.
Excellent question. We typically don't see it. And in fact, in many cases, we see the opposite, where, we design our experiments to really compare motor performance with and without stimulation. And using proper designs, we randomize the order of testing. And what we often see is that, if we do the initial test without stimulation, then do a few trials with stimulation, and then repeat that baseline test without stimulation, we'll see that they're better, that their baseline has improved after even short bouts of stimulation. So, so that tells us, or at least my interpretation of that is that, plasticity is occurring somewhat rapidly. I know that's a bit of a loaded term, but there's clear adaptation happening that is changing their level of spared function fairly quickly.
Okay, great. So we have no more questions at the moment. So I just want to remind that our participants may still post questions into the chat, and please vote for best questions as well. Thank you, Duke, for the great talk. Thank you so much.
Thanks for the invitation.
And thank you. And we have Fan Lam. I'm very pleased to have Fan with us because he represents University of Urbana-Champaign, University of Illinois at Urbana-Champaign. That's university where I had my first Fulbright, and keep warm memories about that. So far I understand Fan also has a lab in Beckman Institute. Am I correct? Oh, that's
Yes.
great. So great place, and Fan is specializing in fMRI studies, more technical side, so far I understand. So Fan, thank you for joining us, and please, you're welcome.
Great. Thanks, Ilya. Can everybody hear me okay? See the screen? Let me just try to. Right, can you hear me okay? And are you seeing the slides?
Yep, we do.
Okay, great. Thank you. All right. Thanks for the kind introduction, and thanks, Ilya, for organizing this this great event. And it's my great pleasure to be here and tell you a little bit about the topics related to our research. And this may be switching gear a little bit. So for our group, this is our group at the University of Illinois Urbana-Champaign, and our group mainly focusing on developing imaging technologies that integrating some of those physics-driven fast acquisition strategies and AI-powered computational techniques to hopefully help understand and study the brain better at the with emphasis on the molecular and biochemical level. And we are fortunate to be generously funded by some of these funding sources this year to support our research effort. And this, as I mentioned earlier, this may be a little bit different from the talks that we have seen today, and trying to develop tools to look at the brain, the processes that underpin brain function and some of the diseases at different time scales compared to the the methodology, neural recording, neurodynamics, recording analysis method that you have seen. So as we know, many technologies have been developed to help us study the brain, covering a wide range of spatial and temporal scales. So I'm lucky to be in a field related to magnetic resonance imaging, shown as MRI, that cover a pretty decent chunk of the spatial temporal scales on this chart here. And as we know, MR-based neuroimaging technologies has been really transformative and help us study the brain, particularly the human brain, by providing us really powerful tools to probe the anatomical, functional organization of the brain, and non-invasively, and particularly in vivo. And we can also use MRI signals to not just to map the structure of the brain, function of the brain, but also use this signal to characterize some of the tissue properties, such as how the tissue interact with the magnetic field, how stiff they are related to biochemical properties and their electromagnetic properties, for example. And in the meantime, we also need to recognize that more than perhaps 95%, and all of these amazing images that I'm showing you here are using MR signals from a single molecule alone in our body. That is the water molecule. And in the meantime, we need to recognize that when we zoom into individual voxel, say in the MR image, we're seeing a tremendous level of molecular and cellular complexities beyond just water molecule. We have different metabolites, neurotransmitters, peptides, lipids, DNA, so on and so forth. And these different molecular processes are implicated in different physiological functions and pathological processes in different neurological, neurodegenerative disease, which are apparently major health issues that are faced by our society, as mentioned by some of the talks earlier. And in the meantime, one thing I would like to note that is these molecular processes also underpin, regulate neural functions across different temporal scales. So we have a very nice suite of tools allow us to study neuronal activity or brain activity as covered by the previous talks. In the meantime, there are these molecular processes such as metabolism and even deeper level genetic activity, epigenetic activities, molecular processes related to gene expressions, for example, they interact and regulate brain functions, perhaps at slower time scales, but also play a very important roles. Excuse me. So one of our goals is to try to figure out, can we develop better tools to help us better study and resolve this molecular level complexity across different spatial temporal scales. And these are the topics I would like to focus on today related to our research. The first one is how our effort on developing what we call multiparametric molecular imaging of the brain using MRI signals. And the other is more recent effort trying to push MRI information to the epigenetic level, combining a few expertise from different disciplines. All right, so the modality of focus today is this particular tool called magnetic resonance spectroscopy imaging, shortened as MRSI. As the name suggests, so it's basically a modality that combines the spatial encoding power of MRI as well as the chemical specificity, the chemical information provided by NMR spectroscopy people have been using in the field of chemistry and biochemistry for decades. And by combining those two, using special physics, computational mechanisms, we can typically produce information of this kind where each voxels, you can obtain spatially resolved spectrum look like this. If you do proton spectroscopy, you have different peaks coming from different molecules or different metabolites in the brain at a particular location. And you can try to detect MR signals from different NMR sensitive nucleus, such as phosphorus-31 and carbon-13. And one nice feature about this is that if you do, for example, proton and phosphorus-31, you can obtain molecular information without having to introduce any radioactive tracer or contrast agents into the human body, into the living animals, for example. So this provides a pretty comprehensive way to allow us to characterize the biochemical profiles of the tissue and map them across the brain in a label-free fashion. And this molecular imaging capabilities, multiplex, I should say, molecular imaging capability have been, shown to have promises in different applications and different studies related to basic neuroscience studies, as well as neurological diseases such as brain tumor, stroke, and epilepsy. And some of the examples are not listed here. In the meantime, the progress of in vivo MRSI has been a little bit slow. It's probably not one of the most popular topics in the entire field of MRI. Although early days, in the 80s and 90s, about 1/3 of the people in the field of MRI are working on MR spectroscopy. But with the popularity of diffusion MRI and functional MRI, and so the attention to this field has been decreased a little bit, mainly due to a few fundamental technical challenges that's faced by the field. First of all, as you can see, because we are not only producing spatial map of the water molecules, now we are adding additional spectral dimensions to the overall imaging problem, right? You are trying to produce this high-dimensional spectrum for maybe one-dimensional, two-dimensional spectrum for every pixels in your image. So it's a higher dimensional imaging problem. And more importantly, this molecule that you're trying to image, detect, and quantify, they typically have 3 to 4 orders of magnitude lower abundance compared to the water molecule, which is really abundant in our body. And in the meantime, there's also these long-standing challenges. Even if you get a nice-looking spectrum that containing signatures from multiple molecules, how do you separate and decompose this molecule to really obtain molecular specific features of individual metabolite to provide more specific information? And apparently that has been really challenging so far. So the reality check is that, if you go to the clinical scan, the tools are typically used in the clinical practice, although not that frequent compared to many of those anatomical and functional MRI exam. You typically get a very poor resolution on the order of 1 centimeter cube. You can imagine a 1 centimeter cube voxels, the tremendous heterogeneity that you are seeing in that individual big chunk of tissues. And mostly you can only do a 2D scan because it's very slow. It's a very high dimensional imaging modality. And then typically takes you more than 15 or 20 minutes to acquire such low resolution data. So what we have been trying to work on is trying to address these fundamental technical challenges, as well as also pushing this MRSI to what we call a multidimensional regime. Why do we want to do that? Let's say, assuming you can obtain a nice looking 1D spectrum from your across the brain for individual voxels in the 3D volume, and you look at this spectrum, you see the very crowded space. The peaks from different molecules, they kind of overlap with each other. So which make it very challenging to separate molecule-specific information. In the meantime, even if you look at this low abundance molecule, there's a huge dynamic range between different metabolites. For example, the abundance of NAA, which is a molecule only produced in the central nervous system by neurons, they are 10 times more abundant than the GABA signal, which is the primary inhibitory neurotransmitter in the brain. In the meantime, you can introduce additional dimension to the MRSI, right? For example, you can do things that you typically do to water molecules, such as introducing T2 weighted scan, diffusion weighted scan. You can obtain multiple spectrum for the same voxel. And why do you want to do this? Because by doing this, it gives you opportunity to better separate different metabolites, and also give you opportunity to encode this biophysical process of different metabolites and to quantify some of the biophysical parameters. Why does this matter? Here's a quick example, hopefully that makes sense. So maybe a lot of you have heard of this modality called diffusion MRI, right? So what it does is encode the stochastic motion of the water molecules inside a particular voxel into your signal, so you can use this pattern of water diffusion in your signal to probe what we call the microstructural information in the brain. But water is just one molecule and it's very ubiquitous. It's both intracellular, extracellular, and equally present in all the cell types. But you really look into each voxel, there are different cellular component and tissue compartment, and the water is basically present everywhere. So this really presents this specificity challenge in terms of probing individual cell types, individual tissue compartment, right? Assuming if you have a way to measure, to quantify the diffusion property of multiple metabolites, not just water, on top of water, then most of these metabolites are intracellular. And some of these metabolites are enriched in specific cell types. For example, NAA is primary in neurons, and some of the metabolites are highly enriched, such as inositol or choline. They are more enriched in glial cell types. So this potentially provide an additional dimension of information to provide intracellular and cell type specific microstructure information. So our effort have been really trying to combine, to develop end-to-end solutions that really combine our understanding of the spin physics, as well as data acquisition strategies, signal processing methods, as well as AI-powered computational method to allow us to really address these technical challenges from an end-to-end fashion. And here's an example that by using such an approach, we're able to produce a technique that allows us simultaneously map multiple metabolites as well as a neurotransmitter, particularly glutamate and GABA, and their biophysical parameters in a single scan across the brain in clinical feasible time. So how is this possible? So one of the underlying driving framework for our approach is this subspace imaging approach that I co-developed with my PhD advisor when I was in my PhD and postdoc period. So the underlying approach assumption for this is that, in the traditional MRI imaging paradigm, MRSI imaging paradigm, we sort of assume every voxel and every spectral point for each voxel are independent unknowns. So you have to acquire that many data points to resolve this tremendous number of independent degree of freedom. Instead of doing that, we recognize in each voxel, the signal basically is a composition of a finite number, a small number of molecules that are actually detectable in vivo. And each one of those molecules has a very unique spectroscopy signatures and determined by a small number of parameters that is experimental and subject dependent. And those MMR spectroscopy signatures has been extensively studied in the chemistry or biochemistry literature. So we can leverage those and then build this what we call a subspace model. The mathematical consequence of this is that if you look at each spectrum, at each voxel, it can be really accurately represented as a linear combination of small number of spectral bases. And if you can use the prior knowledge that we have accumulated by decades of physics and biophysical studies and chemistry studies, then you basically turn these imaging problems from recovering a very high dimensional image function into recover this very low dimensional spatial dependent coefficients. To give an example, if you use a conventional Fourier imaging paradigm, the degree of freedom that you have is basically a number of voxels times the FID point, which can be typically in the order of hundreds. And if you use the subspace imaging approach, it's reduced the number of voxels times number of order, basically is the rank, the order of the low dimensional subspace, which is typically in the order of tens. So then you are significantly reducing the degree of freedom and potentially allowing more flexible trade-off between speed resolution and SNR. So we took this step a little bit further, and instead of having this subspace model for one-dimensional spectrum, we extended to multidimensional MRSI and having this low-dimensional subspace structure, considering both the FID dimension as well as the additional parametric dimension. I promise this is the only slides with the equation. The point to make here is that you can, by using this framework, you can tailor the model to specific problem of your interest or what we call a specific task. For example, if your problem for a particular disease is really trying to separate the major metabolite as well as some of the neurotransmitter signals, then you can tailor your model, for example, to decompose this overall signal into 3 components, one capturing the major metabolites such as NNA, creatine, choline, the other 2 capturing, for example, the inhibitory neurotransmitter GABA and the excitatory neurotransmitter glutamate. And you can optimize your acquisition and reconstruction in this particular task-specific context. And how do we learn this low-dimensional subspace? And here's a quick illustration of how we do it by combining physics-driven modeling as well as data-driven machine learning. So because we know physically, through decades of chemistry, physical literature, we know ideal situation what the chemical structure should look like for individual metabolite. So then we can simulate a large number of this realistically looking NMR spectrum, and by sampling parameters collected from literature for our own experimental study and feed it into this realistic biophysical model and simulate a large quantity of NMR spectrum. And then you can also use some experimental data to further fine-tune these variations to build this low-dimensional subspace. And we also developed this acquisition strategy to allow us to rapidly generate multidimensional MRSI encoding, combining some sparse sampling theory, and also introducing by combining water acquisition and MRSI acquisition in a single scan. So you can basically acquire some of the information from water molecule in the meantime that can also serve as additional information, also help your data processing, as shown some of the case here, and without having to acquire additional scan, which is really lengthen the acquisition time. Okay, so by doing all this, so this is the result that you have seen. I think we are very excited about this, and we're working with different groups to translate this into clinical populations. And one of the example is post-traumatic epilepsy. Apparently, we learned that about 20% of TBI patients will likely develop epilepsy during the 5 years period post-initial injury. And so one of the challenges or the unmet clinical needs is that it's very difficult to predict the risk for individual developing epilepsy at the early stage. If we could do that, then we can potentially use more a proactive management strategy to reduce the risk of this patient developing epilepsy, for example, giving anti-seizure medication in earlier stage instead of later. So here are some data, pilot data we obtained from a couple of patients. In this case, you can see these lesions in the traditional clinical MRI, and this is in the chronic phase of this injury. You can see this scar tissues, lesions form in the patient's brain, and they are actually sometimes multiple lesions due to a single injury, and they look morphologically somewhat similar. But then if you look at the biochemical information provided by our technology, you can see that there's a significant imbalance for some of the lesions in terms of the abundance of glutamate and GABA. So there's an imbalance of excitatory and inhibitory neurotransmission. And this is the first time we're able to see this in this type of patient. And this is another patient that's showing that this imbalance in some of the lesions, you see a more reduction of inhibitory neurotransmitters GABA than the excitatory neurotransmitter glutamate. So it seems to indicate the promise of this to kind of better understand why some tissues become more epileptogenic than the other tissues. Our hope is to combine this with some of the other imaging features through a multimodal approach to hopefully provide a way to better predict which individual is likely to develop seizure and epilepsy and enable a more proactive management strategy to improve the quality of life. And we can also do this introduce other parametric dimension into this by using our approach. In this case, you can combine diffusion with MRSI. Hopefully the earlier example help motivate this. Why do we want to why do we want to combine diffusion and MRSI? By mapping molecule-specific diffusion parameters hoping to produce a more cell type, a tissue compartment specific microstructural information that allows to better connect these potential markers to specific pathology associated with the specific cell type, for example, in tumor and post-stroke recovery. So it's easier said than done. You can simply combine diffusion encoding as shown here by adding diffusion encoding as people typically do for diffusion MRI with MRI acquisition, because we can do things fast. We can do this, but if you simply combine them, this is the image you're basically seeing. Because MRSI, in essence, without getting into technical detail, is a more extreme fashion of multi-shot diffusion, which is extremely sensitive to motion artifact or any type of system perfection. So by our technique, this is the image you are able to see. So if I show you this image, you probably won't believe me. There's a brain underneath all these artifacts, right? By combining special acquisition and reconstruction strategies, we're able to really address this artifact issue and make diffusion with the MRSI more realistic. And here's an example, one of the recent paper that was just accepted, and showing the capability of producing simultaneous metabolite mapping as well as metabolite specific diffusion coefficient mapping across a 3D volume of the brain and in a clinical feasible time. So hopefully this could be a useful tool and adding some additional a richer set of microstructural biomarker on top of the amazing capability offered by diffusion MRI, and to allow us to study some of the cell-specific pathologies in different neurological and neurodegenerative diseases. All right, and just to mention that you can combine AI power computational strategy to further improve the reconstruction of this high-dimensional spectral function that you are interested in, as shown here. By combining this subspace approach with some deep learning strategies, you can really improve some of the reconstruction for particularly the weaker metabolite signal. In this case, another patient data really showing the improved quality enabled by combining subspace imaging and AI-based reconstruction. All right, so building on this effort, so we are also trying to think about, is it possible to push the molecular level information we are able to probe in the human brain in vivo to another level, particularly coming back this idea of different molecular processes regulate, underpin, and interact with neural activities across different temporal scales. There's a metabolism, which we just talked about. There's also gene expression and a very important molecular mechanism called epigenetics that involve, basically it's a molecular mechanism that underpin the regulated gene expression activity, which turned out to be a longer time scale mechanism that interact as well as regulate biochemical process as well as the neurodynamics measured by, for example, fMRI and EEG. And one of the primary epigenetic mechanism is DNA methylation. And the question here is it possible to push the neuroimaging information further down to the epigenetic level? And this is really a team effort. We have a very, very outstanding group of expertise that coming from different disciplines. And the project is really initiated by Dr. Jun Robinson, who is a prominent neuroscientist who have been studying epigenetics for a long time, and he really interested in, is it possible to probe this in vivo? It can really provide a transformative tool to understand the neural mechanism, both the shorter time scale, as measured by some of the neurodynamics probing tools, and the more longer time mechanisms such as gene expression. Yeah, and then this work has been enabled by a couple of different generations of postdocs and bring us to the current stage. And if you look in the literatures, we are not the first trying to connect neuroimaging data from MRI to epigenetics. But if you look at those papers, what they are trying to do is pretty much measure the DNA methylation or some kind of gene expression activities in the peripheral sample using blood samples or saliva. and correlate that with some of the neuroimaging data as produced by anatomical MRI or functional MRI. So what we try to do is to try to think about, is there a way to more directly probe DNA methylation in the brain? And the approach that we settled is to use stable isotope to label methylated DNA, because this is a very longer time scale process. So radioactive labeling is very limited in this sense because the radioisotope, the decay away, so you are not able to probe this activity across a longer time scale, which is the period that is epigenetic activity try to regulate the brain function, right? And using one of the most effective tools to study stable isotope for isotope detection, which is MR spectroscopy. And we really go back to this metabolic pathway involving methionine as the primary methyl group donor of DNA methylation, and design a strategy using C13 enriched methionine to eventually label methylated cytosine. And we started with a chemistry study to really demonstrate that if we could do this in the ideal world, then is the C13 labeled DNA methylation does give us a unique signatures compared to other methyl group or other carbon-13 signals in the DNA. And we show that, and we identify the frequency range for this particular signature that we are looking for, and before we even design an in vivo study. And here's our first proof of principle using pig as a translational model because they have a large brain. So hopefully at the first trial, we can have sufficient signal because C13 MRSI is a well-known low sensitivity imaging tool. And so the way we did it is we design a complete non-invasive labeling strategy, just feed the C13 enriched diet, particularly all the methionine here, which is an essential amino acid, is replaced by C13 methionine. So the methyl group in the methionine is replaced by a C13. And then we basically feed this to the pig for like 10 to 32 days. And then this methionine will pass blood-brain barrier, convert into C13 SAM, and donate the methyl group eventually to the DNA. And then to validate this idea, so after a certain period, we'll take the brain out and we'll cut it in half and take some sample from one half to do LC mass spectrometry analysis to see that if we are really enriching the C13 methyl group in the brain. And then we also take the other half to do a more non-invasive imaging study to see if we can actually non-invasive detect this type of signal. And this is some of the labeling study. We do show that, after a certain labeling period, we can actually significantly enrich the C13 percentage in the C13, the methyl group in the methylated DNA. Another thing about this is basically C13 is basically chemically the same compared to the C12, which is the most abundant carbon isotope in our body. So you shouldn't affect the biochemistry related to DNA methylation. And then we also show that the labeling actually outpaced the brain volume growth. So this is very interesting. I don't have time to go into this, but it seems to indicate some of the this labeling is underpinned by other mechanism than just volume growth, right? And then we also show that we can actually detect these C13 signatures non-invasively in the brain using C13 NMR. And then we show that there's a significant increase of the signal that we are looking for coming from the methyl group, specifically from the methylated DNA, if we have a sufficient amount of labeling. So this is, I believe, the first, non-invasive map of DNA methylation, although it's in the pig brain. But we do show that there's a regional differences. By leveraging some of the MRSI techniques that I described in the earlier slides, with a little bit of computational SNR enhancement strategies, we're able to show that there's indeed region-specific enrichment of methylated DNA, and our technology, shortened as EMRI, stands for epigenetic MRI, is allow us to resolve these regional differences as summarized by the graph on the right. So there's a regional differences in terms of DNA methylation, as well as the DNA methylation that can be probed by our technology. And then we also try this on a bigger pig because we want to eventually translate this to human. And pig, as it seems to be a very nice translational model because there are similarity in terms of the brain structure as well as the metabolic processes compared to the human. And so we try this, a new postdoc, Chang, design a new feeding strategy. Instead of completely replacing the methionine, we basically design a supplement strategy. So basically, when you try to do human, you can basically take this C13 methionine as a supplement, and then many people are already doing that as a matter of fact. And you can basically feed this to the bigger pig, in this case, like 50, 60 kilo pigs, similar to adult human. And then we also shown that after 20 days above, if you only feed it for 10 days, you don't see significant enrichment because these pigs are not in the rapid growing period. But if you feed it long enough, you do see substantial enrichment. So we are labeling the pool of the DNA methylation that are actively turning over. And here's a very preliminary result that we are very excited about. So if you just take the brain, the entire brain, and then put it into an NMR, ultra high field MRI scanner, and you measure the C13 spectroscopy data, you do see a significant increase of the label brain compared to the control, which is basically the pig that fed with the non-C13 enriched diet. You see a significant increase of the C13 signal that are of particular interest to us, particularly at this particular peak here. So this indicates the promises of potentially we can do this in vivo. So this is what we are really actively pursuing towards the goal of eventually we can adapt this tool to image brain DNA methylation non-invasively in the human brain. Of course, we need to go to ultra high field MRI system because the intrinsic low sensitivity of carbon-13. All right, so hopefully this give you a taste of some of our research and help you hopefully appreciate why we want to develop a tool to probe the molecular biochemical information beyond the typical type of structural or functional information provided by existing MRI imaging methods. So coming back to this diagram, this is something I want to conclude with and to share you some of my thoughts. So we are very used to look at this diagram comparing different technology in terms of spatial temporal scales. But one thing I think this diagram didn't show is the type of information that you are obtaining from different imaging modality. For example, if you look at MRI, right? So most of the square here referring to the structural or functional MRI method that are more widely used and appreciated by people. But if you look at other modality of submodality or tools within the MRI realm, for example, MRSI, you're really, although the resolution is typically lower than anatomical and functional MRI, but you do provide an additional dimension of information, that is the biochemical and molecular level information that cannot be simply obtained by measuring the behavior of the water molecule alone. So maybe this biochemical information could provide a very unique window to probe the brain at a multi-scale, multiple spatial and temporal scale, and provide a new window to perhaps a new type of information to perhaps allow us to connect the information across multiple scale. Because in a sense, the biochemistry does not change whether it's inside a cell or inside a particular tissue. It's basically the same molecule doing similar reaction. Although the interpretation of them may be different, but they are in a way more invariant to different spatial temporal scales. With that, I appreciate your attention and happy to take any questions. Sorry if I have taken a little bit more time than 25 minutes.
Great. Thank you so much. So, frankly saying, that's the first time I hear about studies which are on the intersection of epigenetics and fMRI. It's a glance into the future, of course. Very inspiring.
Yeah, it's still in the early phase, but hopefully this can get us somewhere. Yeah. Yeah.
Okay, so do you have any questions? So currently, maybe someone will post a question. So I would like to ask you. So I've noticed that you reported the data on 3 Tesla fMRI machine, and if currently, so the science shifts to more powerful machines with higher resolution, like 7 Tesla, and so far I remember in UIUC they were experimenting or just considering building some kind of 11 Tesla machine. So, for example, if shifting from 3 Tesla to 7 Tesla, it gives you better spatial resolution. However, it also results in a more huge amount of data and again in more sources of noise. So is there some kind of optimal power of fMRI machine for this kind of studies, or 3 Tesla is enough, or 7 Tesla and 11 Tesla is a better option?
Yeah, that's a very important question, right? I think generally speaking, if you go to a higher field, you do enjoy higher sensitivity, particularly for MRSI. Hopefully, I made it clear that we are very SNR limited because the signal that you're detecting are much weaker than water molecule. So it does enjoy some special benefit by going into higher field. So assuming everything works, and so I mean, theoretically, it's really beneficial, I think. And engineering wise, there are very unique challenges when you go to 7T. I can't say for every other submodality within the MRI realm for sure, but for MRSI, so it's more sensitive, for example, to field inhomogeneity. If you go to a 7T, for example, even you breathe a little bit, the magnetic field in your brain can actually change. In 3T, you rarely notice this, but in 7T and above, you do notice this. So then, guess what gives you the chemical specific information along the spectral domain? It's also field inhomogeneity. So now you are dealing with a field inhomogeneity that could potentially significantly degrade your data. And there are other challenges, of course, like a wider spectral bandwidth. So it does require more dedicated engineering design. So basically, it's hard to take one 3D method that works really well as 3T. It can be, and then you just take it and then change some parameters and run it on a 7T scanner. And can it work really well? I don't believe so. You just need to do like a very special engineering design to overcome some of these challenges that we discussed, and to really make sure that you can take the benefit from the ultra high field, particularly the increased sensitivity. Yeah, and one of the challenges I think also is that if you are looking for, I guess, widespread clinical translations, if you want your methodology, because to be honest, MRSI, I think it's very powerful because it really provides a unique dimension of information that the standard clinical MRI cannot provide. But if you want it to be used in the clinics, you do have to worry about, how many ultra high field systems are actually out there and that people can afford and can then run it, in a more push button fashion. So I think that is one of the gap that the community needs to address in order to, but then in terms of answering some of, I guess, the basic neuroscience questions, the basic questions, it could be a very, very powerful tool. Yeah. Hope that makes sense.
Yeah, sure. Great, and thank you for so elaborated answer. Sorry. So I do not see any other questions, but in this case, I would like to ask one more question, if you're not against. So you was talking about epigenetics in terms mainly of DNA methylation. However, there is a huge field in the epigenetics, to my knowledge. So you wasn't mentioning. I'm talking about microRNA. So a lot of epigenetic studies usually target microRNA molecules. So as a main regulators of transcription and translation processes. So maybe, of course, it's just a huge field, and maybe it's better to focus for you on DNA methylation. Maybe it's just easier, but can you give some perspectives on combining fMRI and studying epigenetics using microRNA analysis?
Right. Yeah. That's a great question. I'm not particularly familiar with the microRNA, but I recognize it's important, epigenetic mechanism and transcription and regulation mechanisms. So as you said, I think what we try to do here is to at least started to open a window to demonstrate that it's even possible to develop an MR-based method that can probe any type of epigenetic mechanism in vivo. So it's not comprehensive, right? Just like fMRI is not a comprehensive measurement of all the aspects of neural activities, but at least hopefully provide a window, but looking into the epigenetic activity in the human brain. And specific to, let's say, they are also, we chose DNA methylation. Why? Because number one, it's one of the primary epigenetic mechanism, and it's also relatively more extensively studied. And number three, well, most importantly, we figure out a way to tag the DNA methylation, and we haven't figured out a way to tag other processes. For example, like histone acetylation is also one of the primary epigenetic mechanism. But we thought about that as well. I think there are a lot of opportunities here. Then we certainly are very open to let's sit together and map out the metabolic pathways and which molecule goes to which and what are specific to this particular processes. And if we could label them, would they generate specific signals that are, excuse me, can we generate a signal that are specific to the things that we are trying to detect, right? For example, like we thought about histone acetylation, but then I don't think it's impossible, but at least so far, we haven't figured out a way to say, if I tag, let's say, the lysine residuals in the histone, that signal will be very specific to the histone acetylation. Or if we tag an acetyl group, that will generate us NMR signatures that are specific to the acetyl group on the histone acetylation. Then back to your question, can we generate a tag and the signatures that are specific to the microRNA, and also make sure that signal has sufficient abundance that can go beyond the detection limit of the current MRI detection mechanism? So I think it's a very exciting topic that we should certainly think about, right? And we do need to come up with a strategy, and this does require a multidisciplinary collaboration. Because I don't think any single group, well, maybe it's possible, but I think it's very difficult to a single group that can come up with this idea. Like in our case, for this particular idea to actually come to this stage, it does require expertise from chemistry, right? As you see in the early validation study, like translational pig models, imaging, and epigenetics, to really put this whole thing together in a sensible way, and then validate it, and then push it to the level that can become a viable imaging approach. Yeah.
Okay, thank you. So we do not have more questions at the moment. So maybe just before finishing our conference, just a short summary. First, thank you all for coming. Maybe if someone has questions to our presenters, feel free now to raise hand and to ask directly. So no need to use chat. Also, I would like to say the words of gratitude to my colleagues in Ukraine who helped me with organizing this mini conference. And of course, a great gratitude to my host in university, Purdue University, my host in lab, and chief of my lab, Sebastian Illia, who actually helped me and supported me in running this event for the first time 3 years ago. And thank you for our great talks. It was very interesting to watch your presentations. Really, I'm very excited with all that. And if someone wants to add something, so you are welcome. Well, I think we can make some summary regarding our small rewards. So we definitely have one leader in questions. So Natalia Evpak has 6 likes on her question she addressed to Douglas Weber. And as for presenters, we have also Douglas Weber, who responded 3 questions. He was asked 3 questions. So to other presenters, if we meet again, so just count on beer on me, or coffee if you prefer coffee. Thank you. Any comments? Maybe someone?
Yeah, I just wanted to thank Illia for organizing this event and appreciate the effort of all Ukrainians and keeping strong. You guys are amazing, and keep on, trying keeping science alive, because better times are ahead. Найкраще.
Sure. We'll stay strong, and Ukraine won't surrender, and our science will grow, will develop with your help, of course. Thank you, guys.
Yeah, thanks again for the invitation. A great event. Yeah, keep in touch, everyone. Yeah.
Yeah, sure.